All right. Well, welcome all to the next third, maybe, installment of our series, what has been an amazing series so far of our EvoFunPath seminars. Sure today will be no different. As a reminder, while the talk is going on, obviously we can mostly only see the shared screen, but afterwards for questions in the round table, we're really trying to encourage a cameras on format, again, to sort of kind of promote our sense of community. So you'll all know each other's beautiful faces for when we see each other eventually in person. So keep that in mind for later on. But today I'm delighted to introduce our speaker, Dr. Tami Lieberman. Dr. Lieberman is an assistant professor at MIT in the Institute of Medical Engineering and Sciences and Department of Civil and Environmental Engineering. Okay. And the Lieberman Lab uses evolutionary theory, as we'll see, as a lens and tool to understand community assembly in the skin microbiome. So Dr. Lieberman has developed techniques for tracking within-person bacterial evolution. This started with her time studying lung infections as a grad student in Harvard's systems biology program. That's where we met and became pals, and then did her postdoctoral training in Eric Alm's lab at MIT. So as you'll see, Tami is not a mycologist until we convert her into being one. We're very excited to have her here today representing the EVO and EvoFunPath, and really looking forward to hearing the research from her lab. Take it away, Tami.
Thank you for being here. It's great to talk to primarily students. It's one of my favorite things to do, so I'm really glad to be here, and excited to talk to you and maybe learn some different perspectives in the conversation from your thoughts on fungi. Okay, so my lab at MIT started about almost 4 years ago now, and we're really interested in how bacteria are evolving within individual microbiomes. And just to make sure we're all on the same page, when people start thinking about microbiomes, they often start thinking, about the sort of species levels descriptions of microbiomes, where you have these relative abundance charts of different bacteria, maybe at the genus, maybe at the species level. And of course, there's interest now going forward in looking beyond that and looking at the strain level and saying, okay, within each of these species, we know that there's some phylogenetic structure, and maybe there's something interesting happening with different members of the species. But at the end of the day, while the underlying structure is phylogenetics, at the strain level, often the description again is still sort of these relative abundances of different strains. And we're interested in all of this, but I think what's different about what we do from a lot of other people is that we even go further than this strain level and really are focusing on how individual bacterial lineages, so the descendants of a single cell, are diversifying and on individual people, and not just looking at describing what's there, but what are the functions of these mutations, and what can we learn about ecosystems and community assembly by looking at these mutations. And the idea that there's just a huge potential, for evolution in our microbiomes every day. This isn't some abstract idea, and we can see this with a really simple back of the envelope calculation. We each have about 10 trillion bacteria in and on our bodies. Each of these bacteria has about 1 million nucleotides of DNA in its genome. And every time one of these base pairs replicates, there's about a 1 in a billion chance of creating a mutation, right? So every time our microbiome replicates, which is like on average, maybe like once a day, maybe nutrient limited, there are billions of mutations upon which selection can act. And so even if the vast majority of these are neutral or deleterious, there's still a strong potential for adaptation. And so we're using these de novo mutations to understand microbiome-based therapies. And you might be saying, well, we study pathogens, why should we care about the microbiome? And I care about the microbiome because I think that there's an enormous potential for microbiome-based therapies. You're probably familiar with the famous efficacy of fecal transplants in curing recurrent Clostridium difficile infections. But this is really just the beginning, right? People have been treated for symptomatic urinary tract infections with an asymptomatic E. coli that outcompetes the pathogen. And I'm really hopeful that there's a future out there for rational probiotics, even for diseases that aren't necessarily caused by the microbiome, but things that might be treated with probiotics, for example, regulating the immune system. And these these probiotics could live on our gut, they could live in our skin, anywhere microbes live in our body. They could be naturally occurring, they could be genetically engineered. I have this dream of bacteria that live on my face that secrete sunscreen whenever I go out into the sun. Yeah, right. But there's a problem really to reaching this future, which is not just identifying or engineering these strains, but understanding the ecology that's necessary to get this long-term colonization. This is the best example I've seen so far of really getting long-term colonization of microbes. And it was a microbe taken from humans, given to humans. And after removal of probiotic administration, we can't detect this probiotic in half of the individuals anymore. And we don't know really what's the difference between these different groups of individuals and really which bacteria have any sort of chance. And the problem is that there's, we're still just in the early days of understanding mechanism in the microbiome, right? We don't know what the niche range is for an individual bacterial strain. So can a particular bacteria that's living right on you right now live on the person in the next Zoom window? Can the bacteria that's living right now on your forehead have any survivability on your chin? We don't know. And we don't know what the niche, the selective forces are that determine these niche ranges. So we know immune system's going to play a role, right? Diet, other microbes. Right? We kind of know what the big classes are, but exactly which factors matter most to which microbes, we don't know. And lastly, we don't know the role of within-person evolution, right? Maybe the microbes within me are already adapted to some unique set of selective forces within me. And so these outside microbes aren't going to stand a chance against these sort of like pre-adapted guys. And what's really cool is that by studying this last phenomenon here, we can also get answers to the other two questions as well, as will become more clear throughout the talk. In particular, what we can know and track from studying mutations is we can understand how bacteria are spreading between people and on individuals, even across body systems. And by looking at what genes are changing, and particularly what genes are mutated over and over again, we can understand what's really challenging to bacterial survival in vivo. And so I did say I'd talk about the microbiome, but really with the first today, what I really want to do is talk about some old work that Rebecca probably knows already, from infectious diseases that really, I think, sets up the power of the tool of tracking within-person evolution for getting fundamental mechanisms. And then I'll talk about applying these concepts to the healthy microbiome. Oops, sorry. And one story from the gut and one story from the skin. Okay, so within-person bacterial evolution has been tracked on a whole genome level since 2006. And it was done in this paper where 2 isolates were taken from the same long-term infection at different periods of time. Their whole genomes were sequenced, but there was still a problem, which was which of these mutations are truly adaptive and which mutations are just happening by chance and hitchhiking along for the ride. And to really understand which mutations are adaptive, if this were a lab experiment, we'd have an answer right away, right? We could inoculate replicate flasks, propagate these bacteria in these flasks for some period of time, take isolates at the end of the experiment, sequence their whole genomes, and look for commonalities in these different flasks. And when we look for these commonalities, we call them parallel evolution, and we infer that these mutations are probably likely to be adaptive. Ideally, we'd like to do this in humans, but of course, that's not really an easy experiment to do to get the same exact strain in many different people. And so back in 2011, what we realized was that an outbreak is kind of like a case of these replicate flasks where each person is starting with a very similar strain. And so we can compare evolution in these different similar people. Similarly, people infected with similar strains. This work was done while I was a postdoc, while I was a graduate student with Rowie Kashoni, along with JB Michel, who was a graduate student in the lab at the same time, and to our 2 wonderful physician collaborators, Alexander McAdam and Greg Prevey. And this outbreak was of a bug that infects people with cystic fibrosis, which is the most common common genetic disease among Europeans, where people get these the buildup of thick mucus in their lung that's hard to clear, and as a consequence, they wind up getting these long-term bacterial infections. And there's been a lot of improvements over the past decades, but the life expectancy is still less than 40. And these long-term bacterial infections can be caused by a variety of different microbes. They're usually thought to be microbes taken from the environment. And usually there's sort of one microbe that's really dominating that person's infection, and we tend to treat the patients, or at least doctors, tend to treat, that dominant bug as the main pathogen for that person. And so usually it's these one-off things from the environment, but sometimes there are these transmissible strains that spread person to person. And there was this outbreak of Burkholderia dolosa in the 90s that was particularly problematic and killed, about half of the 40 patients that infected. What we liked about what we thought, this was a terrible outbreak, but it was a really good study case, because as this weird outbreak was happening in the 90s, the physicians were going in and storing clinical isolates. And so we were able to go into the freezer and pull out 112 isolates spanning 16 years from 14 patients. Again, since we study the microbiome, I just like to be clear that isolates is a single colony isogenic clone. Okay, so the first thing we can do is ask how fast these bacteria are evolving. And if we just plot the number of mutations each bacteria has against when we first isolated that, we get this nice linear accumulation of about 2 mutations per genome per year. Okay, but of course, we'd like to know more about what these mutations say. And so we can build a phylogeny of how these isolates are related. And here, isolates are colored by which patient they came from. So what do we see? We see some patients' isolates generally cluster together. And so for each patient's diversity, we can infer a most recent common ancestor. This is the node in the tree that's closest to the outside, from which all those patients' isolates are descended. So what we see is that some patients' isolates, some patients' most recent common ancestors look like they're descended from other patients' most recent common ancestors. Right? Their diversity looks like a subset of other people's diversity. And so we can use this to generate a transmission network, right, where we can say that it's likely that patient N had the bacteria that infected patient N had an ancestor in patient H. And while we didn't sample everyone in this outbreak, we were able to confirm that this outbreak was person-to-person spread and not from some hospital source. But of course, we want to know, how are these mutations changing? What are they actually doing to the bacteria? And so the first thing we can do is, of course, just go into the lab and measure how they're changing without any genomic information. And so we measured resistance to antibiotics, because these patients are just doused in antibiotics. All of these patients have seen ciprofloxacin. And we can see that there's, so this is the same tree I've shown you before, but it's just flattened out now. And we can see that resistance to ciprofloxacin varies over orders of magnitude across these isolates. And we look at the genomic information and see how this informs that. We can see that almost all of this resistance can be explained by mutations in the well-known target of ciprofloxacin, JARE. Maybe that's not so surprising, but it's kind of interesting, right, that you see the same exact mutation popping up over and over again. And even we see very similar mutations popping up within the same patient. I thought I had added some slides. But of course, we're very biased there, and there may be adaptive forces that we're not expecting, right, that we'd like to learn without any preconceived notions. And so we reasoned that we could use this pattern of multiple mutations to identify genes that are under selection in a systematic way. And under a neutral model, there's only about 500 mutations in this whole outbreak. And so we'd expect some genes to be mutated twice, but no genes to be mutated 3 or more times. We find many genes that are mutated 3 or more times, and some mutated like many times than that. And so these genes aren't just chemically more likely to get mutated, they're really driving selection. And we can see this by looking at the canonical measure of selection, DNDS, where high values of DNDS suggest a pressure to change the amino acid sequence, right? They're non-synonymous. So this is a ratio of amino acid changing mutations to amino acid preserving mutations. And so higher values suggest a pressure to change the amino acid sequence, and lower values suggest a pressure to stay the same. So these genes that are mutated 3 or more times show this enormous signature to change the amino acid sequence, while genes mutated twice or once do not. Interestingly, if we didn't segregate genes by how many times they were mutated, we would have inferred a neutrally evolving epidemic. What does this tell us? By looking at what the genes that are mutated 3 or more times, we can get an idea of what the bacteria finds as challenging to survival in vivo. And so we get genes that are involved in antibiotic resistance, genes that are involved in how the bacteria look on the outside, which might have something to do with immune evasion. But there's a number of genes that were unexpected including 3 here that were all involved in the same oxygen-dependent gene regulation pathway. And this pathway hadn't before been implicated in lung pathogenesis. And, but it's just this evidence really suggested it strongly. And our collaborators have since went on to show that this pathway does indeed regulate phenotypes associated with virulence. We're not really still sure exactly what they're trying to do, though, why they're trying to change this 2-component system. So this is an oxygen sensor. Why they're trying to change this oxygen sensor. You know, there were 17 mutations in this one gene. But all 12 of those 17 were just in these 2 small domains. So they're not just trying to break this gene, right? They're trying to do something specific and tweak it, but we don't know exactly what. Okay, so hopefully from the story, what you've seen is that by tracking within person evolution, we can infer something about how bacteria have been transmitting in the recent past. And we can get an idea of what bacteria care about in vivo, in this case, changing how they're responding to oxygen. And so given that the power of tracking recent evolution, you might ask yourself, how come there haven't been like lots of studies by now tracking within person evolution in the microbiome? There's this obviously enormous potential. The microbiome field's been hot for a decade. And I think there's a bunch of reasons. And I think one is a conceptual reason. The first reason is that people think that if you want to study within person evolution in a new cohort, you have to have this beautiful collection of isolates collected over time, and or have a long PhD where you have to first wait to collect, you know, these really long time series. And I thought so too. I was quoted in the popular press saying such a thing, that, you know, this was only possible because of our great collaborators, which it was. Our collaborators were great. But in many ways, I was wrong, because we can actually track within person evolution or infer recent within person evolution by looking at the coexisting diversity at a single time point within an individual person. And I didn't appreciate this at the time because I didn't appreciate how much diversity really coexists within an infection or colonization. Right? We could imagine a scenario where there's always some most fit bacteria, and that most fit bacteria replaces all of its ancestors and its cousins and competitors. Alternatively, bacteria with adaptive mutations could rise in frequency in the population, but because no one genotype is necessarily better than another, they could coexist for a long period of time. Or maybe because they're separated in space, or maybe they're slightly functionally different, right? But for some reason or another, they could coexist. And we think that the second model is true for a lot of different bacteria, both in health and disease. And my animations got a little screwed up. And we know this from some work I did on the same infection, where we took sputum from surviving patients, and instead of just looking at one colony, we sequenced many individual colonies from the same sample. And this is just 29 colonies from one subject, and we can see that there's quite a lot of diversity here. The scale bar here is about 20 mutations. And so there's all these different lineages that are separated by about 20 mutations. And if you combine that with this molecular clock date I showed you before of 2 mutations per genome per year, we can infer that these coexisting lineages have an ancestor about 10 years prior to when the sample was collected, right? Which is also matches pretty well with when this patient was known to be first colonized. Even further, I'm just showing you one gene here, but we can identify parallel evolution from just looking at these coexisting lineages, right? Where there's the same gene. This is one gene, PG, the antibiotic resistance gene, and I'm sorry about the alignment here. But this gene was mutated on this branch of the lineage, on this branch of the tree, on this branch of the tree, and on this branch of the tree, really in parallel. So we can identify that just from one time point. Okay, and so to say this another way, you know, adaptive mutations are rising in frequency, and they're removing their ancestors, but none of them is completely taking over. And this coexistence of adaptive lineages is allowing us to project back into the past and moving us into a single sample paradigm. Okay, so that's reason number one. I think, and then the second reason that within person evolution, the microbiome hasn't been studied that much is because of technical reasons. In the microbiome field, as you may be familiar with, there's sort of 2 main approaches. One is 16S rRNA sequencing, which gives you a sort of species level barcode of which bacteria are there, which of course doesn't provide enough resolution to really understand mutations. But then there's a technical fundamental limitations of metagenomics, right? In theory, metagenomics can tell us everything. In theory, if we just sequence everything, right, and just chop up all the little bits of DNA, and maybe we can paste them back together and figure out everything that happened. But in reality, this is actually really too hard of a problem for looking at within-person evolution at the kind of tree level we're looking for. What we want to identify are these de novo mutations, these coexisting polymorphisms within a sample. But you can have coexisting polymorphisms within a sample, not coming from de novo mutations, but from co-colonization from very different lineages infecting that same person. Moreover, even if you can remove these pre-existing variants and just look at de novo mutations, it's impossible to know which de novo mutations are in the same cell. And so you're not going to be able to make these trees, which will really enable powerful inferences. I just want to make an analogy here, right? It's like, imagine that you have your grants folder or your applications folder on your computer, and you chop it up into little bits. It's going to be hard to know which typos came from the same exact draft, and especially when there's horizontal gene transfer of paragraphs from one application to another. Okay, so sorry about my silly analogy. Okay, so what do we do instead to get around this? We do a type of single cell sequencing, but it's just old school, where we just plate on Petri dishes, and because each colony is formed by a single cell, we kind of think about it as an isogenic clone. Of course, mutations happen during culturing, but those can usually be computationally filtered out. Then we do evolutionary reconstruction, and we focus on these mutations that happened on the tips of the tree only. Of course, this sounds really expensive and laborious. It's not really that bad. We got it down to less than 20 bucks. We're getting close to less than 10 bucks per colony. And so you can really imagine doing quite a lot of colonies from an individual. Of course, the downside is you need to focus on a single species. Okay, so the first question we want to talk about today in the gut microbiome is, do any of these mutations even matter in the gut microbiome? In the cystic fibrosis lung, it's maybe not so surprising that you can knock out a transcriptional regulator or change a transcriptional regulator, because these bugs are coming from soil and now they need to survive in lungs. And maybe there's lots of little tweaks you can make and do a lot better. But we don't always see adaptive evolution when we look at bacterial infections. And in this study we did in Mycobacterium tuberculosis, we didn't, in patients who were not treated with antibiotics, we didn't see any strong evidence for within-person adaptive evolution. And we reasoned it was because these bacteria had been experiencing hosts for a really long period of time, right? And so what we know from theory and from experiments is that as bacteria are experiencing the same environment, the rate of adaptation tends to slow down, where really adaptive mutations are exhausted early on, and then there tends to be less opportunity to do much better by just simple point mutations. And so maybe this, the CF infections are down here, and maybe our gut microbes should be over here, because as we know, our microbes have been diversifying with us, with mammals, for at least a long period of time. And so this work was done while I was a postdoc with Eric Alm, in collaboration with Jay Zhao, a graduate student in the lab. And this work is on a particular common gut bug called Bacteroides fragilis that lives in the large intestine, and we just focused on it because it's common and easy to culture. We focused on really healthy subjects, so we're not going to see any antibiotic resistance evolution here. They're so healthy that their poop is literally medicine. Their poop is being sent to people to treat C. diff. And from each of these subjects, we took stool samples from some just one, and from some of these longitudinal samples up to a period of 2 years. And from every sample, we sequenced about 20 to 40 colonies. So this is a tree of just one person, their Bacteroides fragilis diversity, and it kind of looks like we saw with Betulosa, where there's a bunch of different coexisting lineages here. And I'm sorry that the scale bar has went away, but this overall distance here, average distance, the tip is about 8 mutations. So it's not, that many mutations. And we also see, but there is some coexisting diversity. And we see here that there's also gains and losses of mobile elements. So these are mobile elements that are present in some colonies and absent in others. In this case, these two are consistent with losses, but we also see other cases that are consistent with gains. And so the first thing we can ask is, how long has this diversity been coexisting? And to do this, we can use the same principle of a molecular clock, and we get a clock rate of about 1 mutation per genome per year, kind of similar to the 2 mutations. And so when we look at this tree here, we can say that this most recent common ancestor was probably about 8 years ago. But these patients aren't 8 years old, and they weren't like first probably colonized by Bacteroides fragilis 8 years ago. These people are in their 20s and their 30s generally. And so this raises the question of whether this person was colonized 8 years ago, or maybe there was some adaptive sweep or some neutral sweep 8 years ago that removed diversity. And we think it's probably that there are some sort of sweeps occasionally, because actually what I kind of didn't point out, what was on the slide the whole time, is that this bottom lineage down here was only captured at the first sampling time point. So if we had started our sampling later, we would have inferred a most recent common ancestor. Right? So we think that maybe there's occasional sweeps. But if we really want to know is, are these sweeps under the pressure of adaptive evolution? And so of course we look for genes that were mutated multiple times and parallel evolution. And we found 16 genes that were mutated 2 or more times within the same sample. And again, these genes that are mutated multiple times show this enormous signature for adaptive evolution, while genes mutated just once do not show the signature. And again, we can kind of get a sense of what these bacteria are experiencing in vivo. And what was striking in this case was that 6 of these genes are all called sus-C genes or sus-D genes. There's one sus-D. Okay. So what's a sus-C or a sus-D gene? There are these importers of complex polysaccharides that sit in the outer membrane, and we don't know which polysaccharide each importer works for, but we do know that we do think that each one is involved in some sort of complex polysaccharide, either from the diet or from the host. What was interesting is that actually one of these genes that was on our list of sus-C genes was actually the strongest hit for essential colonization factors in a mouse model. So they did a transposon mutagenesis study, and this gene was essential for colonization in a mouse model, but the same gene is getting point mutations in individual people and trying to change. Right? So on long, and what we see is that if we look at DNDS in these same genes on long time scales, we see a signature of purifying selection. So in long time scales, there's this pressure to stay the same, but in short time scales, there's this pressure to change the amino acid sequence within individuals. So what's going on, right? Why is there still on this ongoing adaptation in these key genes, right? Another one of those genes was like essential for capsule formation. Well, there could be some new selective forces that are new to the human environment, but maybe there's some selective forces that are only in certain individuals or only at certain time points or certain locations within individuals. We see evidences of like kind of all of these cases where we see evidences of things that look person specific, where these genes are mutated 3 times in one subject and no times in other subjects. And we also see cases where we can't really figure it out, but they look like more common selective forces, right? Where the same exact amino acid is happening in 3 different subjects. We see some cases where it does look like there may be coexisting competing selective forces and pressure to diversify within an individual. And we're lucky enough we can do this because we're lucky enough that not only do we have isolate data, but we also have a very frequent time points from metagenomics from the same subject. So we have metagenomic sequencing every other day for 2 years. And so we can take how we know the mutations are connected from the isolates and combine that with the mutational frequency from the metagenomics and create this diagram of how these bacteria were evolving in vivo. And so we can say is that prior to the start of our study, there were this diversification into these 2 different coexisting sublineages, probably about 8 years prior to sampling. The number winds up being the same as that other subject. And then after that diversification, for this, at least during the whole 2-year time periods for which we have time points, these 2 lineages continue to persist and coexist. And they do that even though there's adaptive dynamics within one of the lineages, right? So this blue guy, this blue genotype is completely whopping its ancestor of the purple genotype, but isn't removing the ability of the orange genotype to coexist and survive. So it suggests that maybe there's some sort of frequency-dependent selection or maybe metabolic diversification. But we can't really figure that out because we don't know if these bacteria are coexisting or maybe they're just separated in space. Okay, so before I move on to the skin microbiome, I just want to just wrap the story up really fast and say that what's really under pressure to change in these bacteria is the outer membrane presentation and the transporters that sit in the outer membrane. We find these contrasting signatures of selection on long and short timescales, suggesting that there's some sort of non-constant selective forces. And really, this is the first evidence of within-person evolution in our gut microbiomes, right? This is the first evidence that I had seen that bacteria within our guts are really adapting on maybe meaningful timescales. And this has some importances for microbiome engineering, right? If each of us has a unique set of selective forces, then maybe we need to select the probiotic strain that's going to have the best competitive ability against the unique set of selective forces within you. But I want to now return to this question of, what's going on with this cool seeming diversification. We'd really like to be able to answer these sort of questions, but it's really hard to do so in the gut where we can't know where bacteria are coming from. And so in my lab, we've really made a shift and we've been focusing on the skin microbiome where we can really understand how geography plays a role in diversification and evolution. The skin is also pretty cool because there's a lot of important diseases that don't have very good animal models. Mouse skin is really different than human skin. And we can ask, we can, and not only is it, and it's, and as I mentioned, it's sort of more tractable. And it's also more tractable for a bunch of different reasons, right? We can think about different spatial scales. We can think about faces and backs and one pore and the next pore. It's not that diverse. And so we can really imagine characterizing how individual species behave and how the community behaves and interactions and really getting a full mechanistic picture. And you may be thinking that it's just sampling from the environment, but your skin microbiome, and in particular your sebaceous microbiome, is really stable over time. It's your microbiome. You have the same strains that are different than the person in the next office. And so yes, the sebaceous microbiome is the microbiome of your face, your back, and your chest, your sort of oily skin. And it's dominated primarily by this one bacteria, Cutibacterium acnes. But things are not that boring. There's actually quite a lot going on because there's many strains of Cutibacterium acnes coexisting on each person. And this is work we know from metagenomic studies from other groups. And so the story I want to talk to you about with probably most of the time remaining is why are these different strains of C. acnes coexisting on the same person? You could imagine that they may be functionally different, or maybe they're just neutrally coexisting and because of some sort of priority effect or geographic distribution. And this is work led by Erin Conwell, a postdoc in my lab. But it was started while I was a postdoc in Eric's lab, but really Erin's carried out the most of it in our lab here. So C. acnes, what is it? It's this anaerobic bacteria that can survive in oxygen, but prefers anaerobic growth. It's really the dominant thing in your pores. It prefers to grow at the bottom of the pores, and it's considered a commensal. So it's really, it's called acne, right? It's got this nasty name, but it's on literally everybody. So it may play some role there, but it's not causing acne on its own. And I'll just mention really quickly that there's some studies that say that certain strains cause acne, and I don't believe them. Okay, so throwing that out. I'm not that political, sorry. Hi, so what did we do? We sampled Cutibacterium acnes from healthy subjects, taking samples from the surface and from individual pores. From each of these samples, we then cultured for Cutibacterium acnes, sequenced whole genomes, and performed phylogenetic inference. So we get high spatial resolution and high genetic resolution. Okay, so first thing we can do, we can recapitulate is the well-known phenomenon that multiple strains are coexisting on each person. And so this is the tree of all the C. acnes we collected here. The scale bar is much bigger than the trees I've been showing you before. And when we just paint on it all the isolates we got from our most heavily studied subject, subject A, we can see that there's these 6 different coexisting lineages on the subject, some of which are actually very close to each other on the evolutionary tree and also have very similar gene content. And so we don't think that they're that functionally different from looking at their genes, but maybe there's more going on. And we see kind of similar patterns across people, where there's many strains, and sometimes many lineages, and sometimes many lineages from the same sort of strain group. So the first thing we can ask is where these strains are spatially segregated, and we don't see dramatic differences on different parts of the body. There's a lot of noise here in these bars because we don't have that many colonies from each area, and so functionally we can't really say that these are that different from each other, at least at this coarse level. And we don't see dramatic differences between the surface and within the pores. Okay, so far nothing. So far they're everywhere. But when we zoom in and we look at the level of individual pores, we do see a striking signal, which is we can ask at the level of individual pores, how likely is it for two colonies to be from the same lineage? And given the distribution of lineages on subject A, we'd expect that about half of the pairs of colonies from pores should be from the same lineage. But what we actually observe is that colonies from the same pore are almost always from the same lineage. Really suggesting that each pore is dominated by its own separate lineage. Right? In other ways, each island kind of your face has its own population. And so what's going on, right? Well, what's going on? Well, first, I think the next question you want to ask is how close are these islands to each other? Like what's the spatial segregation here at this finer resolution? And to do this, we can take one of the samples we took from the pore strips. And what we can see is that there really doesn't seem to be a strong spatial segregation, right? You can see here that these three pores that were, within a couple millimeters of each other, were each, I got 3 colonies. We got 3 colonies from one lineage from this pore, 2 colonies from another lineage from this pore, 3 colonies from another lineage from this pore, really different lineages really nearby. So we can ask, are these pores selecting for different lineages, right? Maybe just this blue pore just prefers blue lineages. And to do this, we can zoom in and look at the diversity within an individual lineage. And so here, this is just a tree of one lineage from our most sampled subject, colored by pore. Okay, and the gray samples are coming from the surface. And so what do we see? We see that pores are dominated by single genotypes, really. So this sort of rules out this idea that there's just filtering, right? Because if there was just filtering, you'd expect that there'd be multiple different genotypes from the same lineage within that pore. But instead, we just see one member of that lineage within that pore. And we see the same pattern when we look at other subjects, where we have very low diversity within a pore, but despite higher diversity in that lineage on that subject. And so you might be asking, okay, and if you look, it's a really very recent common ancestor too, right? This low diversity. There's on average less than 1 mutation per cell within that pore. So just that the population within that pore is really young and it hasn't been in that pore long enough to on average accumulate any mutations. And so we can ask, is this because of population bottlenecks, or just selective sweeps? And we really don't think it's selective sweeps, because if it was selective sweeps, we would see strong signatures for adaptation, right? There would be adaptive events within each pore selecting for its own unique genotype, right? And if that would happen, most of the mutations should be, adaptive, or a lot of them. And we don't see any signature for adaptive evolution. Genome-wide, everything looks purifying. And if we segregate genes by how many times they're mutated, we again just see purifying. You have to trust me, I've looked every possible way for adaptation I could in Ireland, and we cannot find it. So so we said, so there's got to be some sort of neutral bottleneck happening, right? That's not adaptive. That's happening within every pore. And this neutral bottlenecking is not some trivial bottlenecking, because these populations are big, right? You can have 10 to the 6 bacteria in one of these blackheads that we're pulling at, right? So something else is going on. And we think these bottlenecks are recent. And I just want to point out another feature of this that you can kind of see in the tree, but we kind of quantified it here, which is that these bottlenecks aren't old. They're not old bottlenecks that happens early on when this person was first, maybe first colonized, and then each pore is kind of separated forever in its own little deme, because we see that pores are kind of related to each other, right? We don't see this comb-like tree structure that you would get if each pore is just colonized once and then separate. And so we think these pore-specific mutations didn't happen within the pore, but probably happened during the colonization event of that pore, where pores are maybe destabilized and then recolonized. And so why would there be a bottlenecking? Well, we think what's going on is that at the bottom of the pore, there's this preferred area of growth. And on the top, the bacteria are not growing very much. And it's hard for the bacteria to get into this area of best growth because there's sebum flow to fight against, and because they're just not, it's hard for them to diffuse against all the way down there, and they don't have good ways of swimming down there. And so once a bacteria, so it's very rare for these bacteria to get to the bottom, and once they get to the bottom, they grow quickly and outcompete and fill up the pore before the next bacteria can come in. That's sort of the idea we have. But I think there's other possible explanations, but what we really can say for sure is that there's neutral bottlenecking at the level of individual pores, and this mechanism is one plausible mechanism. And we think this intrapore priority effect may explain the coexistence of these different pores, different lineages on individual people, because these bacteria are not competing with other strains. They're really having just sort of these neutral bottlenecking events, where on the surface it's well mixed, but on the surface there's not a lot of growth, and so most of the growth is happening in this area where there's not a lot of competition. So we think this is one possible explanation why C. are coexisting, but of course there may be other explanations as well. Sorry, just this little graphic here is just to show that maybe this purple strain is much worse at growing on the face, but it doesn't matter. It's got its own food. All right, so we see is that genotypes are spatially segregated across pores. We don't see strong signature for adaptive evolution for C. acnes. And there's a strong intrapore priority effect, which may explain, limit competition, explain this coexistence. And we think this is kind of cool because it's a case where like the actual physical structure of the skin can maybe promote coexistence and change sort of ecological and evolutionary trajectories. And going forward in terms of probiotic therapy and implications, what it says is like this priority effect is so strong. And so that's the thing we have to focus on for C. acnes if we want to engineer probiotics. It's not necessarily about picking the best strain, probably, but about removing the strains that are already there and exposing a new niche. And so I think for the sake of time, because I want to have questions, I'm not going to tell you this other story about the skin in detail. I'm just going to flip through it really fast and just say that not everything on the skin is neutral and boring, and that that in the context of eczema, which is atopic dermatitis, where you have itchy patches of skin colonized by the bacteria Staphylococcus aureus, we see rapid adaptation of Staphylococcus aureus on kids with atopic dermatitis, where we see these sweeps where mutations emerge and then sweep over the entire body into different sampling sites on pretty short timescales. And these mutations, we've identified at least one of these targets of this mutation. I don't know what's happening. I'm stuck. As capsule loss. For some reason, on these individuals with atopic dermatitis, they're rapidly losing their capsule, and losing this protective coating is for some reason giving them a survival advantage. We're still trying to figure out. Okay, so with that, I just want to say that we a couple conclusions. We see that colony sequencing is powerful for getting in vivo insights. That in the gut microbiome, there's still ongoing adaptation even during health without antibiotics that we don't understand what the reasoning is for, and it's an exciting area. And that on the skin, we at least see one case where the structure of the environment can change the ecological and evolutionary trajectories. Going forward, I think there's a lot of open questions in this area of within-person evolution that are really exciting for students to explore in the future. When we see one case of adaptive evolution and one case of neutral evolution, And I showed you one more case of adaptive evolution at the end. And what's balancing, what's determining these forces, right? What are the rules? Is it just spatial structure? Is it phylogenetics? Is it phages? Is it other microbes? We don't know. When we see these mutations happening on an individual host, are they going to make the bacteria better in general, or are they just sort of local dead ends? What's this role in changing the ecology and ecological dynamics? Big question. We're not answering in one project. And you know, of course, the question is, what's the role of these mutations when we see adaptations? How do they impact disease? Okay. So I mentioned that these the key players of this work, but all of this work was really highly collaborative, and there were many individuals involved in all of these works. And with that, oh yeah, I'll just mention that we're always looking for great postdocs. So you know, the US and Boston, good spot to be. This is Mount Auburn Cemetery in Cambridge. Get a good view of Boston. And I'll stop and I'll take questions.
Oh, wonderful. Thank you.
Thank you for your attention.